Howdy guys, my name is Sofia Guerra. Today I will be going over Lab Video Report 7 with my lab partner, Mayra Landeros, which is Biochemical Test Part 1. For our introduction, the purpose of performing biochemical tests on bacteria is to identify and differentiate bacterial species based on their metabolic capabilities. Each test detects a specific biochemical reaction that reflects enzyme activity or substrate utilization. The gram-negative unknown organism 285 was tested on to determine its metabolic profile and assist in identification among similar species. The indole test detects the ability to break down tryptophan into indole using the enzyme tryptophanase. The methyl red MR and Voges Proscar test determine the type of fermentation pathway utilized, mixed acid or butanediol fermentation, respectively. The citrate test determines whether an organism can use citrate as its sole carbon source. For materials for the media used, we had tryptophan broth for indole test, MRVP broth for methyl red, and Vogue's Proscar test, and Simmons citrate agar for citrate test. For the cultures tested, we had E. coli, E. cloacae, and our unknown 285, which was a gram-negative. For procedures, for the indole test, we inoculated sterile tryptophan broth tubes with each organism and incubated at 37 Celsius for 24 to 28 hours. After incubation, 0.5 milliliters of Kovacs reagent was added. A red ring indicates a positive result, which is the presence of indole. No color change indicates a negative result. For MRVP tests, inoculated MRVP broth tubes and incubated at 37 Celsius for 24 to 28 hours. For MR, we added 1 or 2 drops of methyl red reagent. A red color indicates a positive mixed acid fermentation result. For VP, we added 15 drops of VP1 and 5 drops of VP2. A pink-red layer indicates positive acetylene production. Citrate test. We inoculated Simon citrate agar slants with a slight touch from a 24-hour culture. Incubated at 35 to 37 Celsius for 18 to 24 hours. A blue color indicates positive utilization of citrate, and green remains negative. For our result for the citrate test, here we have E. coli, unknown 285, and E. cloacae. First of all, what this test is trying to show, or what we can see from this results, is that the organism uses citrate as its only carbon source. So if there's a color change, the meaning behind it is that blue indicates positive, which means alkalization, and green remains negative. And here we can see that both our E. coli and E. cloacae um, changed to a deeper greenish color, which means or indicates negative, no citrate was used. But here in the middle, we have a blue unknown 285, which indicates a positive result. So overall, we can see that our unknown utilized citrate as a sole carbon source.
Now for our indole test, we can see here that there is a clear separation in all of the test tubes. Some look more cloudy than others. Our principle here was to detect the ability to produce indole from tryptophan via tryptophanase. And our Kovac reagent, the way we tell that was if a red layer indicates that it's positive. As you can see here for our unknown 285, we see no change in color, but we do notice that split in layers and a little cloudiness to the tube. So therefore, negative. As for our E. coli, it also showed a separation between layers with a clear ring but no color change to it, indicating that it's also negative. For our E. cloacae, same result, no change in color, but there is a separation between layers, indicating that is negative. Overall, none of the tested organisms produced indole, indicating lack of tryptophanase activity. Now for our methyl red MR test, our principle was to detect stable acid production from glucose fermentation, which is a mixed acid pathway. Our reagent was methyl red color being positive and yellow being negative. For our E. coli, we see that there is red layer that stayed on top, indicating a positive, strong acid production. For our E. clocae, we can also see that there is a red layer at the top, indicating positive. As for our unknown 285, we also see that there is a red layer at the top, indicating that it's also positive. And overall, E. coli performs mixed acid fermentation as well as the other two. Now for our Vogue's cross-core VP test, our principle was to detect acetone production from the butanediol fermentation pathway. Our reagents were VP1 and VP2. Pink/red equals positive. For our E. clocae, we can see it's a cloud deeper and more pronounced, equal positive. For our unknown 285, same thing as the E. clocae, indicating that it's positive. Now for our E. coli, it is more on the grayish towards the top of the tube, which can indicate it being negative. In relation to the MR and VP are complementary. Organisms positive for MR are typically negative for VP and vice versa. This fits with the observed results. In conclusion, the biochemical test successfully differentiated metabolic traits of E. coli, E. clocae, and the gram-negative unknown, which is our unknown number 285. E. coli was endo, MR positive, VP negative, citrate negative, indicating mixed acid fermentation typical of the species. E. clocae was endo negative, MR negative, VP positive, citrate negative, consistent with butanediol fermentation. Unknown 285 showed endo negative, MR negative, VP positive, citrate negative, suggesting it may belong to the Enterobacter genus. These tests demonstrate how biochemical reactions serve as key diagnostic tools in bacterial identification. Thank you.